Hi everyone. In this video, we are continuing with 5.4 non-Mendelian genetics and the clues to recognize what they are from a chi-square. So we've talked about several other reasons why you would reject a null hypothesis. And in this video, I'm going to talk about lethal genes. So lethality is also sometimes called fatality, and that's mostly by me. It is an inheritance pattern in which a genotype is lethal. So in other words, the combination of two particular traits ends up with a lethal combination, and all the offspring that inherit those specific characteristics end up dying. So the example that we have here is coat coloring for mice. And in this particular coloring, you have a very common color called agouti, which is kind of like a very dark gray, brown, very neutral color. You can see here that when an organism is homozygous dominant, they end up with that agouti coat. Well, in the case of agouti, what happens is that if the organism inherits a particular variant or a different type, so we're using the same letter, but we're using a superscript, what you could end up with, so this would be a heterozygote, what you could end up with is a yellow coat, and that doesn't affect the color of these rodents. However, when you get the two variants, and again, the variant just has a superscript Y to indicate that it's different from this one, two combinations of this ends up being lethal because of the gene interaction. Now, most of the time, what's going to happen is for this lethality is that the organisms are going to end up with a homozygous condition. And that homozygous condition is we're going to prevent the organism from forming properly. In other words, going through embryonic development. So how are you going to know if you reject a null hypothesis due to lethality? So you will know it's lethality when one phenotype is completely missing from the offspring. Now, this is not from one individual test. This will be after many offspring produced. And what you'll notice is that even though your Punnett square predicted one of these certain phenotypes, when you look at the actual offspring, 0 of them are coming out to be one of the predicted phenotypes, and this may be after hundreds of offspring produced. And so, if that phenotype is completely missing from the offspring, then when you reject the null hypothesis, you could argue that because of the missing offspring, because of the missing phenotype in the offspring, this particular inheritance pattern could have been due to lethality. And then that is why it doesn't follow the traditional Mendelian genetics. I hope this video was helpful. Thanks for watching. Bye.